whole food web. And that response is likely to involve mechanisms operating at different levels of ecological organization, beginning with the organism level, a single plant, which can go through phenotypic changes. These are fast changes in the behavior of the plant and different traits that are changed on the short time scale. One example you all know, plants that plants in shade that grow towards light. Another example, which is relevant to what I'll be talking here, is stomatal closure as a result of water stress. Stomata are these microscopic openings in the leaves, and through which the plant exchanges gases with the environment. It takes in carbon dioxide, but it also releases water. And under conditions of water stress, to keep water levels, it closes the stomata. At the population level, meaning many plants of a given species, but also of other species, we can have vegetation patterning. This is an example of bands or stripes of acacia trees in Australia. No one planted those trees in that pattern. And that pattern is a way to cope with water stress, because those trees that survived now can draw water from the berthal surrounding. And this is an extra source of water that compensates for the reduced rainfall. At the community level, which may be few or many populations of different species, community reassembly can take place. For example, shifts from fast-growing, intolerant species to slow-growing, stress-tolerant species. And these mechanisms interact in complex ways. And the goal of our research is to try to disentangle these complex responses using mathematical modeling. So I'll start with the models we develop and use, and beginning with modeling phenotypic changes. And here I'll motivate a general equation for phenotype dynamics using stomatal closure as a case study. Then I'll proceed to modeling vegetation pattern formation and coupling it to phenotype dynamics, and then modeling pattern forming plant communities, where I'll be using a trait-based approach. I'll explain later what do we mean by that. And then using this model, we'll study various response forms, and I'll gradually increase the complexity, but start from the simpler cases of phenotypic changes without vegetation patterning. This may be relevant to ecosystems where the feedbacks that form spatial patterns are too weak. Then in that case, I'll describe the bifurcation structure of steady states and the implication in particular to plant communication about impending or potential stress. Then I'll consider vegetation patterning without phenotypic changes, again showing the bifurcation structure of steady, uniform, and at this time also pattern solutions, periodic and localized. And I'll discuss applications to basically two specific ecosystems. And then I'll consider the more complicated case of vegetation patterning and phenotypic changes, and discuss an example of phenomena that cannot be explained by any response alone, any of the two responses alone. And finally, I'll consider vegetation patterning and community reassembly. And here, I hope I'll have enough time for that. And here, I'll show, according to the model studies, that spatial patterning can buffer community structure. And then I'll conclude and try to share with you my view that a unified theory of ecosystem response to climate change is something that might be feasible, which is related to similarity in bifurcation structure that we see for different mechanisms. Okay, so let's begin with modeling phenotypic changes. There are many, many types of phenotypic changes. And the question that we ask, is there a universal equation describing them and what this equation may look like? So again, this is research in progress. We work out a particular example, stomatal closure, and then we'll try to generalize it. So stomata are those little macroscopic openings in the leaves. There are, as you see, there are many of them. They consist of pairs of banana-shaped cells called guard cells, which contract and close the stomata when the turgor pressure inside those cells is reduced. The closure of the stomata reduces water loss by transpiration. Transpiration is the process of the plant taking up water from the soil through the roots and eventually releasing it to the atmosphere through the stomata. And this is, closing the stomata is a very significant way to cope with water, or to prevent dehydration, because more than 95% of the water that is taken by the roots is released back to the atmosphere. Now, in modeling phenotypic changes, or stomatal closure in particular, we want to capture empirical observations. One of them is hysteresis. Stomata remain partially closed after the stress is released. And cueing. Stressed plants can communicate cues to nearby plants and elicit closure of the stomata of those unstressed plants. Okay, so stomatal closure is a very, very complicated process involving a network of biochemical reactions and physical processes. This is a simplified scheme. It begins with water stress detection by sensors in the cell membranes, which trigger the opening of ion channels and the influx of calcium ions from the apoplast, this is the extracellular space, into the cell. That actually also trigger influx of calcium ions from internal resources, from the vacuole, which is a big vesicle inside the cell. Those calcium ions, when they accumulate in the cytoplasm, they bind to protein kinase and activate it. Protein kinase catalyzes the transfer of a phosphate group from ATP to a target molecule. In that case, the target molecules are a transcription factor for gene expression for the synthesis, eventually for the synthesis of the hormone abscisic acid, or ABA in short. That ABA is then transformed through xylem sap to the leaves, but it also feeds back positively on calcium ion influx, forming a positive feedback loop that accelerates this process. The ABA that reaches the leaves induces an outflux of osmolyte solutes from the guard cell, thereby increasing the water potential in those cells and induce outflux of water from the cells, which leads to reduce the turgor pressure and cause contraction and closure. So this is a summary of much more complicated processes, but this still, to too complicated for our purposes, modeling purposes, and also too specific to stomatal closure. So we want to abstract that scheme into two steps. One is stress detection and signaling, which we quantify by a state variable psi, which in this case can be associated with the concentration of calcium ions. And the second step is the phenotypic change itself, stomatal closure, which we quantify by another variable, phi, which in this case can be related to the ABA concentrations. And more specifically, we define psi and phi to be dimensionless variables. Here the subscript mid represent the midpoint between the maximal and minimal concentrations of the relevant agent. So according to this, phi negative represents intolerant phenotypes, phi positive tolerant phenotypes, but it's not a binary situation. We have a whole continuum of phenotypes from intolerant to tolerant. Now for psi, the signaling variable, it grows in time. This is the way we model it. It grows in time because of water stress. So SW, the soil water content, drop below the characteristic value, W start. Psi start increasing, meaning in that case, the concentration of calcium ions. The concentration of calcium ion is also increased by ABA. This is the positive feedback that I just discussed. We write it as some function beta of phi. We don't know that function. We expand it in Taylor series, keeping up to third order, going up to third order, choosing the third cubical to be negative to truncate at that order. And then we have another term, minus gamma psi, which represents the fact that as calcium ions accumulate, they become toxic to the cell. So there is a process that leads to their decay. For the phenotypic change, well, it's triggered by the calcium ions. The ABA synthesis is triggered by calcium ion. This is the first term. It's being reduced by catabolism, the breakup of ABA molecules when their concentration increases. And ABA leaks to the soil, from the plant roots to the soil, and can diffuse. And so these are the two equations that we have that we propose to use in describing stomatal closure. But these equations are not closed in closed form because they depend on the soil water content. And the soil water content is affected by biomass growth. There's more water uptake. And biomass growth, in turn, is affected by the phenotypic changes. So we need to couple these equations to a model for the biomass and water. And we want that model to capture vegetation patterning if we want to address not only phenotypic changes, but also spatial patterning as stress adaptation mechanisms. So vegetation pattern formation is driven by positive water vegetation feedback loops. It's a feedback between local vegetation growth and water transport towards the growth location. With this feedback in mind, we can imagine a uniform vegetation, like grassland. And now we decrease precipitation. And if we, by chance, have a patch of denser vegetation, according to this feedback loop, that patch will draw more water from its surrounding than the surrounding does. So there will be net transport of water to that patch, which will increase its density, but at the same time decrease the density in the surrounding. And so this is a so-called scale-dependent feedback. It has positive effect on short scales and negative effect on larger scales. And now we may ask, how does vegetation draw water from its neighborhood? There are several mechanisms. One is overland water flow in crusted soils. It turns out that the soil in drylands tends to be covered by soil crust. This can be physical crust, just a denser top layer because of the impact of the rain, but quite often these are biogenic crusts. For example, cyanobacteria that forms a thin, few millimeter thick crusts. Those crusts do not develop, so they first reduce the infiltration rate of surface water into the soil, and they don't develop in vegetation patches. For example, cyanobacteria need sunlight, which is blocked by the litter of the vegetation. So the overall effect is that there is a infiltration contrast low in bare soil and higher in vegetation patches. The fact that the root zone also makes the soil more porous, and that further increases the infiltration rate in vegetation patches. And this contrast in filtration rate induces overland water flow to the vegetation patch. The vegetation patches actually act as sinks for water flow. And this mechanism, this is an example of a system in Australia, grassland gap patterns, nearly hexagonal. They are pretty big, as you can see here. In that case, 4 meters in diameter on average. Another mechanism is soil water diffusion. The roots take up water, deplete the water content, induce soil water gradient, and soil water diffusion. This, an example of that mechanism appears to be grassland gap patterns in Namibia, the so-called fairy circles. If you heard about that, they are also big, actually can be even bigger than in Australia. And there's yet another water transfer mechanism. It's water conduction by laterally spread roots. And here's an example of a system in Trinidad Island. It's a tiny island east of Brazil, Brazilian island. And what you see here, there are 2 species in that system, but the green patches are woody vegetation, Willandina bondug, and it has extremely long roots. Here you can see a root exposed, and it goes all the way from the green patch into the grass here. And actually, in this process, we have that woody vegetation displaces the grass. Okay, so we want to model those feedbacks and 3 water transfer mechanism, and this is a model that does that. The overland water flow is captured by the water flux of surface water, the soil water diffusion term in the water equation. So H is the surface water, W is soil water, and B is biomass. And the conduction by lateral roots is captured by the non-local behavior of this growth rate, GB, which is given by this form. And what it says is that the growth of plant at the point X depends on the availability of water at point X prime, which the roots of that plant extend to. And the roots architecture is captured by this kernel function. Okay, so now I want to consider, for the sake of simplicity, a single scale-dependent feedback. We choose to be, I choose to be the saltwater diffusion, which applies to a system with sandy soil. In sandy soil, the infiltration rates are high everywhere, so there is hardly overland water flow, and we can actually eliminate the equation for the overland water flow, H, and left with two equations. And then the roots are, I want to consider confined roots, and therefore I can get rid of those integral of the laterally spread roots. This gives rise to this algebraic factors where E is the root to shoot ratio. And now in these equations, I want to couple them to phenotypic changes. And well, we know that when the plant changes its phenotype from intolerant to tolerant, meaning increasing the variable phi, then the plant becomes more tolerant, so its mortality decreases, but it grows more slowly also because the growth rate, the growth rate decreases as well. This is because as stomata closure, the plant can tolerate water stress, but it doesn't take as much carbon dioxide, which is needed for photosynthesis and growth. So growth is slowed down. So we have 4 equations that are in closed form and we can study, but we can do a simplification because the signaling process, psi, occurs on a much faster time scales than all other variables. So we can eliminate it adiabatically, set the time derivative to 0, solve for psi in terms of phi and w, plug it in the equation for phi, and get this equation, this equation for the phenotypic change, which captures the two phenotypes, negative branch for the intolerant one and positive branch for the tolerant phenotype. So now we have a closed form 3-variable model capturing both phenotypic changes and spatial patterning. In general, all the other coefficients that appear here, eta 1, eta 2, eta 3, can also depend on the soil water content and may also, with different phenotypic changes, may change sign as W decreases. And I'll come to that at the end of the talk. So let's now we want to study this model, and I want to consider phenotypic changes without vegetation patterning. This is the first case we consider. So, this, which means the scale-dependent feedback, the form patterns is too weak to form patterns, and we have basically spatially uniform solutions. And this is the bifurcation structure for that case. So we have the bare soil solution, which become unstable at some precipitation. We have the intolerant phenotype that disappear here in the solid node bifurcation, and the tolerant phenotype. And there are a few insights that this bifurcation structure, the diagram, tells us. First, potential resilience. We see the tolerant phenotype extends to lower precipitation than the intolerant one. So this increases resilience to drought. It tells about tipping. Unlike the classical case of tipping from a functional state to a dysfunctional state, here we have the plant has a choice. Once the intolerant phenotype ceases to exist, to either tip to bare soil or to go what we call pathway of resilience to another functional state, the tolerant state. And in that case, we may have rate-induced tipping if the phenotypic change is too slow compared to the precipitation downshift and the biomass response, then the plant finds itself in harsh conditions when it's still intolerant, and then we have tipping to bare soil. Another thing that this bifurcation diagram reveals is that we may have biostability and hysteresis, and biostability of the two phenotypes, tolerant and intolerant. And this is related to the positive feedback of ABA on calcium ion influx. You can see from that equation that if W is larger than W star, so we don't have much stress and the intolerant phenotype is stable, if we increase sufficiently phi, we may shift the system to the tolerant phenotype. Then if phi is large enough, it may be bigger in absolute value than the first term, and the phi will increase. Okay, so and another insight is plant communication in the biostability range by propagating ABA cues. And here I want to describe a beautiful experiment that has been done by my colleague, Ariel Novoplansky. He also, there's a nice TED talk by him, Plant Learning, that I recommend to listen to. In that experiment, they took a series of pots and planted plants so that each plant has roots in two adjacent pots, okay, as illustrated here. And they impose stress on the first pot. They can do it by reducing water or by increasing salinity. It's similar effect. And so the first plant closes stomata, okay, while the others that are not subjected to stress kept the stomata open, but in the course of time, the second plant closes stomata as well, and then the third, and then the fourth. So what is happening here? This is a reduced form of the mechanism. We have stress detection, influx that leads to influx of calcium ions, that leads to ABA synthesis and stomatal closure. But we also have this positive feedback that ABA has on the calcium ions. And this is the response of a stressed plant. But if ABA has a positive effect on calcium ions, also external ABA will have a positive effect on calcium ion and eventually lead to stomatal closure, even of an unstressed plant. So this is the response of an unstressed plant that is exposed to ABA. And that ABA may come from the leakage of ABA from a stressed plant, or not necessarily this plant, a plant that responds by stomatal closure, leakage of ABA to the soil, and the next plant can sense it and respond as if it's subjected to stress, although it is not. So this explains this wavefront or queuing wavefront that has been observed. And now we want to see whether the model reproduces those queuing wavefronts and what else can we learn from it. So indeed it reproduces. This is a specified time plots of numerical solutions of the model equation that show this wavefront, but provides a few new insights. For example, that we may have at higher precipitation also the queuing front that propagate in the opposite direction. This is not surprising from the pattern formation point of view. We know that there is usually a Maxwell point when we have fronts between two spatially uniform states. Another insight is that by decreasing the fitness of the tolerant phenotype through the functions of the growth rate and mortality on the phenotype variable phi, the system may become excitable because we also have separation of timescales, and then we can have pulses of instead of fronts, where the leading front is the queuing front and the trailing front is the queuing front. And these are still need to be tested experimentally. Let me proceed to vegetation pattern without phenotypic changes. And then it turns out most models have a similar bifurcation structure, and this is a typical bifurcation structure for vegetation pattern formation. We have the bare soil solution here with zero biomass, which is destabilized by forming uniform vegetation, but uniform vegetation is stable only at high precipitation, and as we go decrease precipitation, it undergoes a Turing bifurcation to periodic pattern. And there are additional solution branches, periodic solution branches as we decrease precipitation. Actually, many of these, here I plotted just two, culminating in a single patch solution in the finite system that we are considering. In addition, there is a biostability range of uniform vegetation and periodic patterns where we have localized patterns. This is the story of homoclinic snaking, whoever knows about that. So we have, in that range, we can have localized patterns, and actually the uppermost branch correspond to uniform vegetation with a single gap, the lowest one to periodic pattern with a single missing gap. And a few insights. Well, this is a self-organization phenomenon. Periodic patterns appear spontaneously below a threshold precipitation, the Turing instability, which explains this type of phenomena. There is multiplicity of stable states, patterns, periodic, localized. There is higher resilience of the pattern states compared to the uniform. And this is because of the extra source of water that vegetation patches draw from the bare soil surrounding. And tipping, there again, like in the phenotypic changes, here even more, there are many pathways of resilience, transitions to periodic patterns of different wavelengths, and also the possibility of tipping to bare soil once we go below the stability range of the uniform vegetation. Another insights come from the two-dimensional bifurcations, from the bifurcation diagram for two-dimensional solutions. Then we have three solution branches bifurcating at the Turing stability, hexagonal gaps, stripes, and hexagonal spot patterns. The gaps, however, are the first to be stable, then stripes, then spots. And altogether, we have a sequence of 5 basic states along the rainfall gradient at high precipitation, uniform vegetation. As we reduce precipitation, we get hexagonal gap patterns, stripes, spots, and then bare soil. And these patterns are observed in nature. So this is kind of encouraging, but yeah. Just a minute.
Sorry. In the plot you were showing before?
Say again?
The plots you were showing before, it seemed that the pattern states have higher biomass than the uniform one.
Well, that depends very much on the metric of the vertical axis. You can choose it to be the maximal biomass, the spatial average, the L2 norm. There might be situations where the biomass doesn't change much because you have less covered space, but those patches receive more water and can grow more. But that depends. I mean.
But in the plot, that was the local maximal biomass, or
this I think was let's see. I think there was L2 norm. No. Here it's maximal biomass, but here is the L2 norm in the 1D. I mean, so this is why it's different in these two diagrams. Okay, so there are many additional, quite many observations that this type of modeling can explain. And I want to focus on two examples. One is what ecologists call fairy cycle birth and death. So the disappearance and the appearance and disappearance of fairy cycles. Fairy cycles are these types of gap of bare soil in otherwise uniform grasslands. This is a picture in Namibia. Now in Namibia, the soil is sandy. So, and the roots are laterally confined. So they cannot, so the overland water flow is not relevant. High infiltration everywhere into the soil on very short timescales. And also the confined roots, which are span a few tenths of centimeters, cannot explain those big fairy circles of 10 meters or even sometimes more. So what is left is the soil water diffusion transfer mechanism. And when we consider a model with that mechanism, which I actually showed you before, we get this bifurcation diagram. Again, this is similar to what we've seen. Here we see in a better way this homoclinic snaking branches of localized patterns. And in particular, a single missing gap. In 2D, this is a single missing gap, and this is observations of a single missing gap. Okay, so this is an example of an area that we identified where there is a birth of a first circle. Okay, so this the time span of close to 10 years. Initially, there is no first circle here, but it appears here after 10 years or so. But there was in between, in 2007, severe droughts. And so these are observations that type of observations that people are talked to. What we did, we took the model, we took the initial distribution of first circles, observed one, as initial conditions for the model simulations. And lo and behold, when we mimicked a drought, and lo and behold, we get a first circle born in, where it was missing here. But in order to get that, we had to start within the homoclinic snaking range, let the drought take us out of that range, and when drought is over, go back to it, but to a different solution branches with more gaps. So this tells us that first cycle birth and death are basically state transitions in the homoclinic snaking range. So this is one type of insight that the model gives us. There is basically no meaning for first cycle death. I mean, it depends on the rainfall regime. Sorry. Okay, oh, it jumps this way. Okay, and another example which I just mentioned very briefly is patchy, this system that I mentioned in Trinidad Island, patchy invasion of woody vegetation into grasslands. The nice aspect is that here we have laterally very long roots. And what you see, this is a kind of blow up, zoom in into the top region here. So you see that the woody shrub, the green patches, Guilandina, displaced the grass. This is in 2019. And you see it compared to 6 years earlier, where the grass was still in that area, not displaced. And this is the models that take into account relevant aspects of that particular system, reproduce that behavior, including the hollows around the vegetation patches and the displacement of the grassland, and additional insights, which I'll skip now. And so let me go now to vegetation patterning and phenotypic changes. As I said, focusing on example where each response alone cannot explain the observed behavior. And let's go back to for a second, Namibia. We discussed this sequence of morphologies in 2 dimensions, gap patterns, stripe patterns, and spot patterns. But in Namibia, we see only gaps. We don't see large-scale spots, not large-scale stripes. Instead, what we see is small-scale patterning within the vegetation matrix, in between the first circles, as precipitation drops. And so the question is what we miss. Well, maybe phenotypic changes. This is a very robust behavior. But then what phenotypic change may apply to this case? So it turns out that if we go in the depth dimension, the soil is in many cases layered. There is a loose top sandy layer, and below it there is a compacted deep soil. And the deep soil is moister because there's a lower evaporation rate in that layer compared to the topsoil. So this suggests hydrotropism as the phenotypic change. Hydrotropism is the ability of plants to grow roots toward the moisture soil volumes. Very much like phototropism, the ability of plants to grow towards light. And so if the deep soil is wetter than the topsoil, we may expect a phenotypic change from shallow-rooted plants to deep-rooted plants. And so we propose for that a 2-layer, 2-phenotype models. This study was before we came up with the phenotypic equations. So instead of introducing a phenotype variable, we distinguish between the 2 phenotypes by assigning each one a different biomass variable. Biomass for deep-rooted plants and for shallow-rooted plants. And in the equations, we have terms that describe transitions from the shallow-rooted to the deep-rooted plants, provided W2, which is the soil water content in the deep soil layer, is higher than W1, the soil content in the top layer. And there is a uniform mixed states of the 2 phenotypes. At high precipitation, it's mostly shallow-rooted plants. At lower precipitation, it's more deep-rooted plants. But at some point, we get a Turing-like bifurcation. But 2 modes grow, one with short wavelengths, one with large wavelengths. And when we look at the eigenvectors, we can associate the short wavelengths with deep-rooted plants and the long wavelength with shallow-rooted plants. And the reason why the deep-rooted plants show the small scale is that those roots experience the compact soil layer where diffusion length is very short. So these are numerical simulations. The top row is in the case where we switch off phenotypic transitions, no phenotypic transitions, and we get the classical sequence of patterns from gap patterns to stripe pattern, gap, stripe, spots. But when we switch on the transition rate from shallow-rooted plants to deep-rooted plants, we can get this behavior, gap patterns, but then multiscale gap patterns, the small-scale patterning, and then at yet lower precipitation, we get ring-like patterns. What is happening is that plants in the matrix die out first, while those in the periphery of the first cycles can survive because those still benefit from the water that draw from the first cycle, the bare soil there, that has more soil water content. And this is observations, and very similar, or what we get is very similar to what is observed. And another result is that if we look at the precipitation at which the big scale spots collapse to bare soil, for the parameters that we used here is 123 millimeters per year, while when we have these phenotypic transitions, the collapse of those rings is at 46 millimeters per year. So the combinations of the two stress adaptation mechanisms, phenotypic changes and spatial patterning, results in significantly higher resilience. And another result here is if we decrease, well, we did here simulation where we decrease precipitation slowly at a constant rate. And now the top layer is what I showed you. And now if we decrease the rate of phenotypic transition, it means that the system may meet lower precipitation, face lower precipitation, while we still have shallow-rooted plants because the phenotypic transition rate is lower, okay? So then we can get large-scale stripes before the dynamic switch onto the small scale of deep-rooted plants and once the phenotypic change took place. And if we further decrease the phenotypic transition rate, we can get large-scale spot patterns, and then again it switches to the small scale, and we get small spots within large spots. And if we increase the phenotypic transition rate, we may not get large-scale patterns at all, only small scale, and these are seen. Okay, sorry, I'll go back here. This is the last topic that I want to address, vegetation patterning and community reassembly. So now we are talking, when we discuss phenotypic changes, we consider a given species. It's one genotype that gives rise to different phenotypes. Now we are considering a community of different plant species, different genotypes. And we want to study its coupling to vegetation patterning. So on one hand, community shift to stress-tolerant species. On the other hand, spatial patterning. Both are mechanisms to cope with water stress. And we use a trait-based approach to community dynamics. This means that we don't follow individual species and all the difference between different species, but rather we focus on a few functional traits that are relevant to the context, water stress in that case, and construct a community by focusing on functional groups of species that share the same values of these selected functional traits, okay? So, and we choose two functional traits, growth rate and mortality rate, and or tolerance. And, and as we said, well, there is a trade-off between these two traits. Those fast-growing species are intolerant, less tolerant than tolerant species, which are going more slowly. So we introduce a trade-off variable called coordinate chi. So that chi equals 0 is the limit of fast-growing species. Chi equals 1, the limit of stress-tolerant, slow-growing species. And we characterize the community by the biomass distribution in this trait axis. As you can see here, this is the local community. It may depend also on physical space. And this is a bifurcation diagram of community states. The vertical axis is chi max, is the peak of this biomass distribution, representing the composition of the community. And what we see here, the green solution branch, is the uniform community. And what we see that as precipitation goes down, the community shifts to higher chi, that is, shifts to stress-tolerant species. But at some point, as precipitation decreases, the Turing instability is hit. And then we have a transition to periodic patterns. Okay, so sorry. Before that, this is the representation of the uniform community in the composed space trait space. So it's uniform in space, but picked in trait space. But when patterns develop, what we see is, if we follow this blue line, which represents the periodic pattern, we see that unlike the uniform community, which shifts to stress-tolerant species as precipitation decreases, the pattern doesn't, pattern community doesn't shift over a wide precipitation range. Compare here point C, and D, chi almost doesn't change. But if we look at the patterns, what we see that in C, the patches are big, in D, they are much smaller. So we have what we call patch thinning, and this kind of plasticity of the patterns basically increase the bare soil areas that surround each vegetation patch, and therefore they increase the, again, extra source of water that compensate for the reduced rainfall. So water availability doesn't change much. But as precipitation further is increased, it decreased at some point. This mechanism of patch thinning is no longer effective, and we have a shift to stress-tolerant species. But then there are other solution branches with longer wavelengths that have lower chi values, and we may have a transitions between from a given wave number to a longer wavelength number by what we call patch dilution, and which again increases the bare soil area around each vegetation patch and therefore keep water availability unchanged. And a nice way to look at it is to calculate the trajectories in the Busse balloon. The Busse balloon is the range, is a plane span by the parameter and wave number, and the Busse balloon is the all stable patterns with all wave numbers that represent stable patterns. And here we encoded the community by the color of the trajectory. So blue is fast growing and red is stress tolerant. And we started at high precipitation, where the species are fast growing, no stress. And then as we decrease precipitation, we have the patch thinning process. It's a horizontal line because we're following a solution branch with a given wave number. So no number of patches don't change. And you see that the blue color occupies a wide precipitation range. But as we approach the boundary of the booster balloon, we have a shift to stress-tolerant species. And when we cross the booster balloon, the pattern becomes unstable and converts to stable patterns, which have longer wavelengths. And therefore, we have a shift back to faster growing species. And again, we have the same procedure. Eventually, the system collapses. So what we have here is that thinning and dilution buffer community structure changes over a wide precipitation range. And this is due to what we call the spatial plasticity of patterns, the fact that patterns easily change, whether the patch size change or the number of patches changes. Okay, let me conclude. Vegetation response to water stress may involve multiple mechanisms of stress adaptation. We discussed phenotypic changes and occurring reference, vegetation patterning and repatterning, transitions to longer wavelength patterns, community shifts to stress-tolerant species. These mechanisms may interact. We saw two examples, phenotypic changes and vegetation patterning, which interact to produce multiscale patterns of higher resilience, and vegetation patterning and community reassembly, which interact to give this behavior of community structure that remains unchanged over wide precipitations ranges. Actually, not only community composition, but also functional diversity of these functional groups. So mathematical models and bifurcation theory are highly instructive in explaining empirical observations and in providing new insights. But I think we can go, or my view at least, is that we can go more than that. Such models may pave the way toward a unified theory of ecosystem response to environmental stresses based on the similarity between bifurcation structure of different species. And let me just say a few words about that. We saw this equation for the phenotypic change, having stomatal closure in mind. But let's consider, say, another phenotypic change mechanism where the first term is negligible and the second term is the one that changes sign as the W drops below W star. And for that case, we get this bifurcation diagram, which differs from the previous one in that the unstressed, intolerant phenotype doesn't disappear in the southern node bifurcation at this point, but rather destabilized, okay? And this bifurcation diagram is basically the same as the one we observe for spatial patterning. Here we have uniform vegetation that is destabilized to periodic patterns. And so if we have the same bifurcation structure, we may expect similar dynamical behaviors. And this is the basis. The more we find same type of bifurcation structures for different stress adaptation mechanisms, no matter what organization level, phenotypic changes at the level of single plane, spatial patterning at community, and maybe at the population level, as we show here, then the results implied by that bifurcation structure is expected to apply to very wide range of ecological contexts. Okay, let me thank my collaborators, former postdoc Jamie and Bidesh, current postdocs Mitchell and Dudra, and my colleagues. And here are a few references related to this particular talk. And thank you for your listening. Maybe too long talk. I now watch the clock.
Couple questions?
Well, this is sort of a random question. So, you know, what's the condition that you don't get the oscillations? I mean, in all the models you have only
stationary patterns.
Right. So is that something that's, could you conceive of an ecological system of this type which would have oscillations?
Well, first oscillations we do get on slope. On the slope,
Well, there you get a drift.
Yes, there's a finite wave number oscillatory instability give to migrating vegetation bands. What is happening that the top part of each band receive runoff from the bursal ahead, which infiltrates and doesn't reach the bottom part of the band, and the bottom part of the band actually lose runoff to the next downhill band. So plants in the top part grow, plants in the bottom part of the band die out. And this is observed, empirically observed. In flat terrain, it's not, we cannot get oscillations under physical conditions. We did try to search for oscillatory damped modes that seasonality, for example, can drive. And we did identify, but it's not a strong effect.
Anybody else? Oh,
so I know this is a little outside of what you were discussing, but I know with looking at stress conditions of liverworts, bryophytes, and there's a connection between the stress that they're when drought conditions and what their microbiome is also at the same time. And the microbiome. Yeah. And I know you discussed a little bit of how the bacteria affect water absorption and stuff like that, but I'm curious, what do you think would happen for these types of plants, these larger, more modern plants, and how they're affecting their microbiome, or if there's a relationship there as well?
Well, it depends. There might be several effects. There are fungi that actually have symbiotic effects and increase the water availability to plants. And there might be microbes that lives on litter that is decomposed. Actually, they may decompose that litter and they produce toxic materials to the plants. And there are models that study that effect. And that leads to traveling waves, because think of a vegetation patch. As it grows, well, in the middle, at some point, the microbiome leads to a higher level of toxic material that leads to dieback, to plant death. But by dispersal or clonal growth, the plant can grow to the surrounding bursal, where the microbiotic community is still small, because there are no plants there. So this leads to expanding rings. And when these rings become large, another ring in between may form. So this is also possible. And rings are widely observed, vegetation rings.